Okay, great. Hi, everyone, and thank you so much for being here this morning. Today, I'm going to tell you about a lot of the work that we've done applying information theory, which Andrea said is another way of inferring higher-order interactions in brain data. So here's a bit of an outline of everything I'm going to talk to you about today, hopefully everything, if I make it through it all. But most importantly, I want to start with why higher-order interactions? Why should you care as a human neuroscience researcher? And if you use networks to study the brain, whether your edges are the structural connections between brain regions or the statistical connections between brain regions, the network formulation is a pairwise approach. The edges are only ever going to connect two brain regions. So I'm interested in how more than two brain regions interact, or a higher order interaction. And I would argue that human neuroscience has actually been interested in these larger interactions for a long time. The resting state networks are a great example of where we think many different brain regions are coming together, and we like to think of those as a system. And these, I don't need to explain to you how important these networks are. There's a lot of ongoing research into mapping them, and they're potentially clinically relevant. So we should really care about these groups of larger interactions. However, even though these are larger sets of brain regions, they're not strictly higher order interactions. And what I mean by that is that they're usually inferred using the structure of bivariate interactions, generally through doing some sort of community detection on a functional connectivity matrix. And they're also not informationally specific. We know that they're interacting in some way, but we don't know exactly how they're sharing information or what way these brain regions are coming together. So this talk is going to sort of follow two themes. First, if we account for truly higher order interactions, not ones that are inferred using bivariate methods, but ones that are inferred using methods that are able to get at higher order interactions, what does the brain's organization look like? And second, how does this organization compare with results from traditional pairwise metrics? And so with that, I'll move to synergy and redundancy. Synergy and redundancy are the two concepts that information theory gives us to describe how larger sets of variables can interact with each other. Redundancy is an interaction that copies the same activity over many units, right? So you can think of a lot of brain regions that are synchronized, or perhaps some are anti-correlated. The point is that the information that they have is the same. And of course, this is very useful for a system like the brain. It's useful for signal amplification, signal propagation, robustness, all sorts of things. We need redundancy for the brain to work. The second type is called synergy. And this is an interaction between several components in which the interaction is irreducible to any of the particular components. If you take one of the components away, that interaction just crumbles, it disappears. So there's information that's accessible only in the joint state. The classic example of this is the exclusive OR gate, but I think the far more intuitive example is you having a group interaction with your friends. That group interaction is definitely not the same than if you had a one-on-one interaction with each of those friends individually. So that's something where the information, the essential bit of it, is contained fully in the group and not in a simpler combination. And so this is useful for the brain for transforming input to output and doing meaningful modification of signals. So rather than just passing signals around, we can change them, we can modify them. There's also, I would argue, three separate lines of evidence that indicate that redundant and synergistic higher order interactions play different roles in the brain. They respond differently to stimulation. Synergy might support higher cognitive functioning, whereas redundancy might be more important for sensory perception. And so this is why I use information theory, is because it provides us this distinction between synergy and redundancy, and there's already a lot of evidence that this distinction is very important. Okay, so I've talked about the concept, but now we need to actually get it out of data. So how do we measure this? There's been many, many ways introduced. This is not even an exhaustive list. But today, I'm only going to talk about the O information. The O information is a heuristic measure. It tells us whether our group of brain regions is synergy or redundancy dominated. It doesn't tell us exactly how the synergy or redundancy is shared, but it's very, very interpretable because of this. So it's combined of two measures, and the first measure is simply high when all the brain regions are synchronized, and it's low when all the brain regions are independent. So that sort of gets at the concept of redundancy that I talked to you about earlier. The second measure is low if all the brain regions are independent, but it's also low if they're all synchronized, and it peaks somewhere in the middle where there's interesting information sharing between brain regions. And so the O information subtracts both of these measures to get this very, very interpretable result that if the O information is positive, we say our brain regions are redundancy dominated, and if it's negative, we say our brain regions are synergy dominated. So we can simply take this measure, we apply it to human bold data. I use the Human Connectome project, 95 subjects, and generally I concatenate all of the subjects into one master time series to avoid undersampling these very higher order interactions. You need a lot of data points, is the point. I can talk more about that in Q&A. Right, so we apply it to a set of brain regions. We then calculate the O information and then just ask simply, is it synergy or redundancy dominated? So with that, I'll move into some results. First, can we find synergistic interactions in the brain? And to do this, we optimized the brain regions that we were looking for synergy, so for negative O information. And here on the y-axis, I'm showing you the O information of our optimized subsets and subset size on the x-axis. So as you can see, for plenty of smaller subset sizes, we were able to find a ton of synergistic groups of brain regions. And we found optimally synergistic subsets between sizes of 8 and 10 regions. However, once you get above subset size 20 or so brain regions, the brain is completely awash in redundancy. We couldn't find any synergistic subsets at all. This is a picture of what they look like. So here I'm showing on the surface of the cortex the participation frequency for each of these brain regions in synergistic subsets. As you can see, it's pretty localized. It's also fairly symmetric across hemispheres. So with this, we can say synergy is prevalent in the cortex. But of course, we care also how does it relate to bivariate functional measures, like functional connectivity, Pearson correlation. How does it relate to functional communities? And so the first thing we did was compare to functional connectivity, right? The most basic measure, we're most familiar with this. And we wanted to ask if brain regions that participate in synergistic subsets are more or less likely to be functionally connected. And so each one of these dots represents a pair of brain regions in our data set, and all the pairs are in this plot. On the y-axis, I'm showing you the co-participation frequency in synergistic subsets, and on the x-axis, I'm showing you their functional connectivity, their Pearson correlation. So as you can see, as your set of brain regions gets more and more likely to participate in synergistic subsets, their functional connectivity trends towards 0. This is really important because it means that if you find a pair of brain regions with really low functional connectivity, they could be independent, which is what we usually assume, or they could be synergistically integrated. And you don't know just by looking at the functional connectivity alone. You have to do the higher order analysis. The second thing we wanted to compare to was the Yao functional systems. So to do this, we sampled different subsets with brain regions coming from a specified number of Yao systems. So for example, we sample one subset with all the brain regions coming from, say, the visual system. And then we sample another subset where we have brain regions from each of the 7 different systems. And so the first thing we found is that the mean O information of sampled subsets becomes more synergistic the greater the number of systems we sample. So this means simply that the subsets we're finding on average are more synergistic when we sample from more systems. We also found that the Yao systems, when taken as their own subset themselves, are themselves redundancy dominated, and those are the red dots in that plot, the O information of each of the Yao systems. And then, most convincingly, we found that the more Yao systems we sampled from, the greater the fraction of synergistic subsets we were able to find in that sample. So all of this comes together to say that Yao functional systems, or traditional systems that we get from bivariate interactions, are able to approximate redundant higher order interactions, but not synergistic ones. And synergistic higher order interactions show us a novel organization that actually tends to combine regions from different functional communities. Okay, so finally, what I've been showing you so far is a very static picture of the brain, but most of us care how the brain changes in time, right? It has ongoing activity. So I wanted to extend these findings in time. And to do this is just a very quick technical note. The O information can be written as an expected value over all points in time. So you can simply remove this expected value, and you get a local value of the O information for every time point. So to examine these in time, we simply, at every single time point, optimize for the most synergistic and the most redundant interaction on that time point. For small subset sizes, you can do an exhaustive calculation. For larger subset sizes, the combinatorics are too big, so you're just running an optimization algorithm independently on every time point. And what this produces is a time series of the nodes that are involved in the most synergistic and the most redundant interaction at every time point. So here you can see in the little schematic, it's a time series of node IDs. So what do those nodes look like? Which brain regions are involved? And this is a bit of a plot twist, but here I'm showing you the most synergistic, the most recurrent synergistic brain regions, and the most recurrent redundant brain regions in the time series of triads. And as you can see, they look exactly the same. We were very surprised to find this, but it turns out that the local O information has a really interesting temporal property, which is that the same subsets experience both the most synergistic and the most redundant moments. So here's an example of what that looks like. In this plot, the pink subset is experiencing these really strong fluctuations away from its baseline. So it's having these very redundant moments, it's having these very synergistic moments. It's a very information-rich subset. Whereas the green subset, its dynamics are very damped, and it's not going to show up as being the most synergistic or the most redundant on really any of these time points. So this is an interesting property, but it leaves us with one question. What is it about the activity of these subsets on a particular time point that makes them synergistic or redundant? Because it has to be about the activity if the subset itself is not changing. And so to do that, I went back to the z-scored time series of just the nodes themselves, and I binarized it about 0. And so what this does is it splits the time series into two communities, or a bipartition, based on the sign of the node z-score. So you have one co-fluctuating positive community and one co-fluctuating negative community. And then I simply count. How many nodes at each point in time in our most synergistic or most redundant subset do we have on each side of the bipartition? And what I find is that synergistic subsets tend to split their brain regions across the bipartition. They have some z-scores with negative sign and some z-scores with positive sign, and they like that to be closer to half and half. Redundant subsets, on the other hand, want all of their brain regions to be on the same side of the bipartition, right? So it doesn't matter which sign they are, but they just want to be on the same side. So here's just a little schematic kind of illustrating this. Synergistic subsets, opposite side z-scores, and redundant subsets, same side. This held true for larger subsets as well. On the previous slide, I was showing you just size 3 and size 4. But on larger subsets, we find exactly the same trend. Synergistic subsets tend to skew toward having their brain regions split by z-scores, and redundant subsets tend to skew toward having them all on the same side. So this same little schematic holds true. Okay, so I've told you a lot. How do I tie this all together? Because at first I said that we have these functional communities and synergistic regions combine them, and then I said, wait, but they're actually the same if you look in time. And the way that I think is best to tie it together is that synergistic subsets, regardless of what timescale you're looking at, always integrate regions from internally redundant communities. But the relevant communities are determined by the timescale. So when you have a long timescale, the relevant communities are going to be your functional systems, the YAO systems, the resting state networks, and the synergistic structure is going to integrate across those systems. If you have a short timescale, then the relevant communities is just the instantaneous bipartition, or perhaps a more sophisticated way of looking at communities. That's also possible. But the point is the relevant communities is the ones that are present on that moment. And so the synergistic structure is going to integrate between them. So to summarize, synergistic subsets tend to combine regions from systems that are internally redundant. And pairwise functional connectivity captures redundant interactions, and so do all of the larger communities that are inferred from pairwise functional connectivity are better at capturing redundant interactions, and synergistic interactions integrate between these communities. Synergy combines different sources of information. It doesn't matter whether those sources are instantaneous or time averaged. And then finally, and I think this is the most important point, synergy is a form of integration that moves away from the past focus on coherence and co-fluctuation. And it gives us something completely novel in terms of statistical brain organization that we really haven't looked at before. So with that, I'll thank you so much for listening. Thank you to all my collaborators. And if you'd like to learn more about the time varying aspect of this, I have a poster on Friday and Saturday at 1609. Please feel free to stop by. Thank you. relate to partial correlation?
Can you repeat the question?
How does O information relate to partial correlation? Do I project enough? Yeah.
Okay. Okay, so yeah, how does O information relate to partial correlation? Good question. And to be honest, I would have to think about that more. I'm not sure off the top of my head. Yeah, let's talk about that later. I can't do math in my brain that fast.
How does subsampling affect results? Could it be that if you have a very large system, it's purely pairwise, and when you look at it, coarse grain, it emerges?
Yeah, this is a great question, and I'm really glad you asked this, because I actually had to take out a slide because my presentation was too long where I talked about this. But in a system, so we're using a parcellation with 200 brain regions. And in a system with 200 brain regions, you have on the order of 10 to the 60th possible higher order interactions. So how you choose to sample all of these interactions really, really matters. And it's impossible, of course, to calculate 10 to the 60th interactions exhaustively. And so it's going to change your results depending on what you want to look at. And I think it's just really important to go about that intelligently and with the question that you want to ask specifically in mind, because the subsampling, picking which brain regions you're going to look at, of course, you won't see the results from the brain regions that you don't look at. So yeah, that's a really important question, and you have to do it carefully.
Yes, great talk, thank you. Have you attempted to replicate some of the synergetic interactions in maybe in an independent HCP sample?
So we actually replicated them in the MICA data set, which the multi-omics, or the micro-scale informed connectivity data set. And all of the results, at least, I haven't done the time varying results yet, but all of the time average results that I showed you in the first section replicate in that data set as well. So totally different collection parameters, everything.